Morning, everybody. Today I'm just going to be talking to you mostly about what I've been doing throughout my PhD so far. I'm going to kind of give you a bit of insight into what goes into characterizing these effector proteins that Grant talked about yesterday. So, recap on what effector proteins actually are and what we think they are. We think that they are, well, we know that they are used by pretty much all plant pathogens to increase their fitness against hosts. So they manipulate the host plant's defenses and also cellular processes to increase their own fitness so that they can reproduce and then box spores and go again in the case of Australasian psidii. So yeah, as Grant mentioned, we've found several of these genes that are expressed during the early stages of infection. And what I'm trying to figure out is what these proteins are and what they're doing. So we have these genes that we've discovered, and what we can do with those is we know that those genes encode proteins. And so those proteins all have an amino acid sequence, and those amino acid sequences are then making structures. And so we can use AI models. So this model is called AlphaFold. It's really, really cool. It basically takes the amino acid sequence of the protein, and then it just predicts what it looks like. And so you get structures that look like this. So this is 1260, the one that Grant talked about yesterday. And as Perry mentioned, there's haplotypes in Australasian psidii. So you get two versions of the same protein that differ slightly. And then we also have 5292 and 10048, which are the other ones that Grant mentioned yesterday. And then this other one here, which I've been working on, which is called 143. And so this is basically what a protein looks like. You have these sort of dotted bits. You can see the arrows and the helices, and then in some cases you get these sort of squiggly lines, which are disordered regions for loops and other such things. And what all of these proteins have in common is this residue called cysteine, which Grant mentioned. Effectors are cysteine rich. They tend to be cysteine rich. And cysteine is an amino acid, which when there is multiple of them in a protein, they form these things called disulfide bonds. And disulfide bonds are one of the strongest bonds found in nature. They're super, super strong. And when you have multiple cysteines in a protein, they form these bonds. And for those bonds to form, they require a special environment. And so when we're trying to produce these proteins, we have to take that into account. So in theory, how do we do this? So we have those genes, and then what we can do is put those genes into a plasmid, and a plasmid is just a circular piece of DNA. And in that, we then have a tag so that we can purify it later on. We also have an antibiotic resistance, so we can select for the bacteria that then have our plasmid in them. And we also have the machinery to be able to overexpress and produce our protein. So we can then transform that usually by heat shock. So you just heat the bacteria to 42 degrees and then they take up this plasmid. And we're using these things called shuffle cells. And shuffle cells are just a special type of E. coli, which are really good at producing disulfide bonded proteins. And then again, like I said, you can select for them and then you can go into culturing. And so we use liters and liters and liters of this stuff called LV, which is just a broth, which has yeast extract, which is food for the bacteria. And then you can grow them up and induce them. As I said, we have the machinery in that plasmid so that we can overexpress the proteins. And then once they've grown fully, we can spin them down, blast them open, and take our protein out through purification. So once we have our cell lysate, we can use this wicked machine called an actor. And we go through these steps where we isolate our protein of interest from all of the other proteins that exist in an E. coli cell. So the first step is a bit of a mouthful, immobilized metal affinity chromatography. And this is why we have the tag on the protein, which actually binds to this column, and then the rest of the proteins from the E. coli will flow out, and then we should be able to elute our protein off, and it should be a lot purer. And then desalt is pretty self-explanatory. It gets rid of the salt. And that allows us to do anion exchange, which is a bit of a clean-up step, which separates molecules based on their charge. And then finally, to finish everything off and to get a super pure protein, we use size exclusion chromatography, which separates proteins based on the size. So that's how it should all work in theory, but what about in practice? So we want to first express the proteins. So on the left here is what an uninduced E. coli looks like. So this is on a gel here. All of those bands are all of the proteins in an E. coli cell. And what you get when you induce overexpression of your protein is these really dark bands, which is your protein being overexpressed by the E. coli. And so what we want to see is bands like that. I've tried over 60 constructs, and to give you a sort of idea on how long this takes, the whole process from start to finish takes about a whole week, basically a Monday to Friday, 9 to 5. And I've done this for over 60 of them multiple times, and only a handful of them actually work. We've tried different things to get them going, but yeah, these are a few that have worked. And it's all well and good being able to express them, but you also want them to be soluble. So when you lyse the cell open, you want the protein to still be there and be happy. You don't really want it to sort of, if it's unhappy in water, it'll just clump together and aggregate. So on the left here is soluble protein. You can see that those dark bands are still there even after we've lysed the cells open, which is a really good sign, which means we can then purify that. But then sometimes you get ones like this and this, and you can see that it's just not in there, which is a bit of a shame, but that happens. And then sometimes it is still there, and you go to purify it all the way through, and you get this, I don't know how well you can see that, but this is basically the protein aggregating. So it's just doubling and then doubling and then doubling and doubling in size and just clumping together. But for ones that are soluble, we can purify them. And so this is the size exclusion trace where we separate them based on size. They both come out as a nice single peak. So this is 1260, haplotypes 1 and 2, and they come out as a nice single peak. And as you can see on the gel, there's no more of those E. coli proteins. There's only our protein left and a nice quantity, which means we can then actually go into characterizing proteins. So the aims for characterizing this protein were to figure out what its biochemical characteristics were. So we want to know how these characteristics are impacting its structure and its interactions, and if the existence of haplotypes is actually making a difference between the two versions of the protein that we have. We also want to know what its structure is. So in the protein biochemistry world, it's very much a structure-based society where everybody wants the structure of their protein. So we want to know what the structures are of these proteins, if they are structured and if they're unstructured. We want to know how the structure is mediating its interaction, so what it's doing. And then, of course, we want to know what they're doing in the plant cell. So what are these interacting with in the plant? Where are they going? What are they doing? Just to refresh on AP 1260, this is the amino acid sequence of the two haplotypes, and what I've highlighted here is the differences between them. So this is where we have two versions of the exact same protein, but there's 10 residues that are different, which gives us these ever so slightly different structures. Probably can't really tell the difference. It's pretty minuscule. You can see in the models here, the alpha-fold models, that it's a little bit more confident in one than the other, and that is probably just due to some of those residue changes. But you can see that we have this tail on the end here, which is super interesting, and it's also predicted to be disordered where that tail is apparently supposed to be. So first of all, biochemical characteristics. So we use this thing called analytical ultracentrifugation, which is where we spin the protein in solution at 250,000 times gravity. And what we can do is measure the absorbance as it sediments. So we can get a really good shape then of its size and its shape. And so what we see here is that the proteins are basically the exact same size, but they differ ever so slightly in their shape. And this FF0 value on the right-hand side here is a measure of shape, and so a value of 1 would be a perfect sphere, but you can see that they're both about 1.5ish, so that they're asymmetrical, which is again what we would expect from those predicted models. And we can also look at the secondary structure, and so what secondary structure is that amino acid sequence then starts to sort of fold in on itself, and you get those sheets and those helices that you see in those predicted structures. And those are what we can see through circular dichroism, which is those secondary structure characteristics bend light differently. And so you can see here the traces of those two haplotypes, and they don't differ too much from each other. But what we can tell from this is that they do both have those helices and they do both have those sheets, but they are primarily unstructured or unfolded. Then we can actually look at the structure, and this is probably my favorite technique. It's called small angle X-ray scattering. So we do this at the Australian synchrotron, which is a massive X-ray facility. And so this is basically where we flow the protein through a capillary and shine an X-ray on it. And then based on the diffraction that you get from that X-ray, you can get information on the protein's structure and shape and flexibility. And so this graph here, there's no pretty pictures, unfortunately. It's all graphs. But this graph here, if this was a perfect bell curve, the protein would be completely globular, a perfect circle, but you would get this really skewed to the right, which is really characteristic of an asymmetrical or elongated protein, which again is lending credence to those AlphaFold models that we produced. The bottom value here is the maximum distance from one side of the particle to the other, and this is measured in angstroms, and 1 angstrom is 0.1 nanometers. And so the value of about 100 angstroms for both of them seems really, really small, but in terms of a really, really small protein, it is quite a big distance, which again is sort of lending credence to those ideas that we might have a tail on the end of it. Then we can also look at its dynamics, its flexibility. So this is a measure of flexibility. If this was a perfect bell curve, again, it would be completely folded. If it's skewed off into the distance, it would be completely unfolded. But this sort of pattern here is really sort of the ideal for a flexible protein. So when you see this sort of pattern here, it's showing that there is flexibility in the protein and that it moves about a bit, or it's partially disordered, which again, that tail on the model sort of tells us. And then what you can do is actually take those models and then predict what they would look like in this experiment, and then you can compare it to your actual experimental data, and you get something that looks like this. And usually I look at my colleagues' ones when they do this, and it perfectly overlines with the experimental data, but you can see that these ones don't particularly perfectly overlay, but they are quite nice on the low end, but not great on the high end. So it just sort of tells us again that, yes, those AlphaFold models are super cool and super good, but they might not necessarily give us the whole picture. And then one of the cool techniques is NMR, nuclear magnetic resonance, and I'm sure you would have all heard of MRI. This is MRI without the I and with an N at the beginning. And so this is basically MRI for atoms. And so each of these peaks on the graph is one nitrogen atom in the protein. And so the protein backbone is made of carbons and nitrogens. And so here we're looking at the hydrogens and the nitrogens. And so this gives us a good view of the protein itself and its structure. And so when you get these really clear peaks on the outside is where you get these nice folded bits of the protein, which are all experiencing a different environment. And then in the middle, you get this clumping where is sort of characteristic of disorder, where then the proteins are all experiencing the exact same thing. So they're basically all just interacting with water. So they're all doing the same thing. And so you get this sort of clumping in the middle. And you can see here, this is the AP1260 heptypes 1 and 2 overlaid. And you can see that they're quite similar, and a lot of those peaks perfectly overlap, but there's also small shifts between them, which is probably due to those differences in the sequence. And then, of course, we want to know what they're up to in the plant cell. And so we did this through a bacteria called agrobacterium, which we can use to, it has a unique ability to hijack the plant cell's production facility, basically, and make our proteins in the plant cell. And so we tag them with a fluorescent protein, and you get cool pictures like this, where you can see the proteins localizing in a plant cell. So this is in Nicotiana bendemiana, which is a model system, not notaceous, I'll add. But you can see here the proteins tagged with fluorescent proteins, and it looks like AP1260 heptypes 1 and 2 are localizing to the nucleus, which is where the DNA is stored. And the endoplasmic reticulum, which is the protein factory. Also a little bit around the cytosol, which is just sort of the space in between. And then really cool, when you co-express them together, you actually see that they go to the same place, which is super interesting. Then for 10948, this one here also looks like it's localized into the nucleus. You can see super clearly the nucleus there, and it also looks like it's going to the plasma membrane a little bit as well, which is super interesting. And then finally, 143. This one here, this folded region in the bottom, the blue region, has actually got homology with cellulose binding domains. And cellulose is a key part of the plant cell wall. And this is looking like it's localizing exactly to the plant cell wall, which is super interesting. So that could infer some kind of function there as well. I don't have images for 5292 because it didn't work very well. Still working on that one. So the next steps for this is to use a technique called co-immunoprecipitation, which is where we can take those fluorescently tagged proteins and then pull them down using an antibody and then see what they're interacting with in the plant cell, which then should tell us, what are these proteins interacting with. And then 3D NMR is where I showed you before we had a hydrogen and a nitrogen, and then this time we add a carbon. And so from that, you can basically get a picture of the entire protein. And those experiments have been run. It sat in the magnet for about 3 weeks. And I have those spectra now, so I will spend the next 3 months working on them. And then it would also be really cool to view where these proteins are localizing in Myrtaceae. And so thank you everybody here who helped with the work, and thank you all for your time.